Okay, this is the beginning of Unit 3. Pardon me. Chapter 7, Unit 3 will only have Chapter 7 and 8. Chapter 7, we're going to start talking about microbial nutrition, how bacteria live in their environment, and how we can watch growth and determine growth of bacteria. Um, this unit is probably one of the most difficult of the units to go over. So I do want to be able to take some time at the beginning of each of the labs that we come together for to answer questions. Maybe take 30 minutes or so for each group or for each lab to be able to go over any questions that you might have, and maybe even take some time to do some visual learning from putting things on the board and going over just kind of a mini lecture over certain material. So as you're going through this PowerPoint and going through and listening to it, be sure that you take time to pause, write down any questions that you have, and we can go from there. Okay, here we go. Microbial nutrition is basically based on the things that bacteria need to grow. Nutrition is essentially a process by which chemical substances, which we would call nutrients, are going to be acquired from the environment. From the environment, and are going to be used for some sort of cellular activity like metabolism, synthesis of cellular components, and etc. All living elements are going to require some source, all living things are going to require some source of these major elements. You can see that there are 12 elements here. These are the major and minor elements. The first 6, carbon, hydrogen, oxygen, phosphorus, potassium, and nitrogen, those are the major elements. There is not an organism out there that doesn't need those 6. Then you have the minor elements, and these are going to be sulfur, calcium, iron, sodium, chlorine, and magnesium. These are going to need in smaller amounts, but the amount is going to vary depending on the microbe. Some organisms need more calcium than others. If it's a spore former, an endospore former, they're going to require more calcium than, say, something that is not a spore former. So each of these elements will be given to the organism based on the organism themselves. We have something called an essential nutrient. This is any substance that must be provided to an organism. It can't synthesize it by itself. For instance, vitamin K is an essential nutrient for the human organism. We get our vitamin K from our environment. We consume it in the foods that we eat. Or we get it from the E. coli in our gut as it releases it from our digestive tract. But we don't make vitamin K on our own. So that would be an essential nutrient. We can divide our nutrients into macronutrients. These are going to be nutrients that require a lot of this particular element. We need these in large quantities because they're going to play some very principal roles in cell structure as well as cell metabolism. Micronutrients, these are the things that we call our trace elements. These are going to be required in smaller amounts, and these are generally going to be involved in things like enzyme function, maintenance of protein structure, things like that, the eukaryotic genetics, those kinds of things. Nutrients, they're going to be processed and transformed into chemicals of the cell after absorption into the cell. Think about when we consume food, we break that food down into its parts and pieces, and the glucose goes off over here, the calcium goes off over here, free carbons or free nitrogens will go off to another place, and they will be transformed and will be used in the cell after they have been absorbed into the cell. So you'll have to go through some sort of metabolism to allow for these nutrients to get into the cell. We can categorize our nutrients based on carbon content. We can either say they are inorganic nutrients, meaning that they have a combination of atoms other than carbon and hydrogen, or they can be organic nutrients, meaning that they contain carbon and hydrogen, and usually are going to be the products of living things. If you think about all these organic, you know, our source of protein tends to be legumes or meat. Those all were living at one time. Our carbohydrates come from grains, pardon me, which are, of course, plants. So we get a lot of these things from living, a lot of these elements or these nutrients from living things. We have a number of sources of these essential nutrients, and let's just start talking about them. Carbon sources. Carbon is probably the one element that the cell needs more than anything else, and the majority of our carbon is going to be involved in normal structure and metabolism of our cells, and the place that we get this carbon is generally going to be from some organic substance. We can base we can classify our organisms based on whether their carbon is organic or whether it is inorganic. If you have an organism that is a heterotroph, that means that you must obtain your carbon from some organic form, meaning that it is going to be nutritionally dependent on other living things. Some bacteria are heterotrophs. Most bacteria are heterotrophs. So they have to obtain their carbon from an organic substance. We are heterotrophs. So again, we are nutritionally dependent on other living organisms. Autotrophs use inorganic carbon, like carbon dioxide, as their carbon source. They are not nutritionally dependent on other living things. As a matter of fact, autotrophs are what we call our producers. So these are the ones that give us the stuff that we need to live. Nitrogen, the main nitrogen source is atmospheric nitrogen, or N2. Another primary source for heterotrophs is going to be proteins or DNA and RNA. These proteins and these nucleic acids are coming from other living organisms that we have consumed or that bacteria have consumed. So these heterotrophs, these organic sources of nitrogen, are going to be in the form of protein, DNA, and RNA. Some bacteria and some algae will also utilize inorganic nitrogen. Basically, what they can do is they can take nitrogen and they can go through a thing called nitrogen fixation, which allows them to convert N2, which is unusable, to a usable form where they can go in and use nitrogen as in this inorganic form. Not very many bacteria can do this. More photosynthetic organisms do this. There's a whole nitrogen cycle that we are not going to be able to take time to go into, but there is a whole portion of it in your textbook if you're interested in knowing how these organisms fix nitrogen. Regardless of the initial form of nitrogen, it has to be converted into ammonia, or NH3. It's only in this form can it be directly combined with carbon to produce amino acids. Sometimes if we have all the parts and pieces, so you can have bacteria that will produce certain amino acids, as well as other compounds. Sometimes it doesn't matter what you've got, you can't synthesize those particular amino acids or those particular compounds, and those are going to be some that we will have to get from the environment, and those again will be what we call essential nutrients. Oxygen sources. Oxygen is a major component in a number of organic compounds, and most of the oxygen that bacteria get is going to be from some organic compound like a carbohydrate. You can also gain oxygen sources in inorganic salts, these things that we call oxides. Those inorganic salts will release oxygen, and these bacteria that can use this inorganic form of oxygen will consume that oxygen as its oxygen source. A good number of bacteria use atmospheric oxygen, just like nitrogen. Pardon me. Oxygen is a diatomic molecule, meaning that it bonds to itself. So you have O2, just like you have N2. And it makes up about 20% of our atmosphere. Matter of fact, I believe it's about 70% of our atmosphere is nitrogen, and very little of it is hydrogen, and very little of it is oxygen. So bacteria can use oxygen, atmospheric oxygen, as an oxygen source. Hydrogen sources, it is a major element in all organic and several inorganic compounds. Remember, the one thing that makes an organic molecule organic is the fact that it has carbon and at least one hydrogen. So your organic molecules are going to be a huge source of hydrogen. If you think about your fatty acids have these very long hydrocarbon chains where you have carbon after carbon after carbon, and each one of those carbons is bonded to at least one, if not two or even three hydrogens. So you have a lot of hydrogens that are going to be available in these organic molecules. You have a number of overlapping roles as far as hydrogen is concerned when you're talking about biochemistry and metabolism. Hydrogen can maintain pH. Remember that acids are, the lower the number pH, the higher the acid, meaning the higher hydrogen ions you have. So if you have an environment that has a very high pH, you can put hydrogen in that atmosphere and it will, or in that environment, and it will bring the pH down to neutral. Or if you need it to go a bit acidic, you can actually move it to the lower end of the pH scale. So hydrogen can maintain pH. It can also form hydrogen bonds. Remember that water molecules bond together using hydrogen bonds. The oxygen of one bonds to the hydrogen of another water molecule through those hydrogen bonds. It also is going to serve as a free energy source in the oxidation and reduction reactions that we have in aerobic respiration. You're going to see when we get into chapter 8 in something called glycolysis in the Krebs cycle that we are going to be moving hydrogens from a substrate to a coenzyme, and those hydrogens are free energy that are going to be converted into ATP. So hydrogens are obviously extremely important in cellular biochemistry. Phosphorus, its main inorganic source of phosphorus is the phosphate ion. We can get this from phosphoric acid, we can get it from rocks or oceanic mineral deposits, but we have some other uses for phosphate, and phosphate is important for us to get, and we unfortunately do get most of it from an inorganic source. There aren't very many organic sources unless you're breaking down nucleic acids, and then you have free phosphates. The uses, they are again key components in nucleic acids, so we need those phosphates or those sources of phosphorus so that we can build nucleic acids. We also need it to build ATP. We need 3 phosphates for every adenosine that we put them together, so we get adenosine triphosphate. This is our molecular energy. Also, we have it in the phospholipids of the cell membrane. Remember that the cell membrane has its hydrophobic fatty acid tails bonded to that hydrophilic region of the fatty acid, which is your phosphate ion, and this gives us the amphipathic nature of the phospholipid of the cell membrane. Most of the sulfur sources are going to be in the environment in some sort of a mineral form. These are essential components, and most of the time we are going to get them in some sort of a vitamin. When we make media for bacteria to grow on, we're going to have to add some sort of sulfur component. A lot of ways that we can do this is through the amino acids methionine and cytosine. They both have within their side chains sulfur elements that can be broken off and used as a source of sulfur. Here's a list of another a number of nutrients that are important for microbial metabolism. You can see here we've got potassium for protein synthesis and membrane function, sodium for cellular transport, calcium for stabilization and endospore formation. Magnesium is a major component in chlorophyll, and it is also a stabilizer of membranes as well as a part of the ribosomes. Iron, this is a component of what we call cytochrome proteins, these proteins that will turn color. Zinc is a regulatory element for a number of eukaryotic functions, particularly in DNA replication and binding factors for the use of enzymes. So these are all going to be very important in a number of different regulatory or metabolic processes. Copper, cobalt, nickel, molybdenum, magnesium, silicon, iodine, and boron are these trace elements. These are going to be found in small amounts in some organisms and not at all in others. Again, it's going to depend primarily on the organism itself. Growth factors are essential organic nutrients, and these are compounds that can't be synthesized by an organism and must be provided as a nutrient, just like we've always learned that an essential nutrient is something we have to get in our diet. So if there are things like amino acids or nitrogen bases or vitamins that can't be synthesized by the cell, then the cell must get it from its food source. For example, you have a number of bacteria that can't synthesize all 20 of the amino acids. If it's going to make proteins, it has to have all 20 of the amino acids. Think about all the words you could not spell if we didn't have the letter E. If we don't have all 26 letters, we can't spell a number of words. If bacteria don't have all 20 amino acids, there are a number of proteins that would never be made. And so these amino acids that can't be synthesized by the cell are going to be these essential amino acids are going to have to be given to the bacteria from their food, whether it's in the laboratory or whether it's in their natural habitat. You have a number of determinants that will decide whether or not, what kind of energy and carbon your organism uses as a source of carbon and a source of energy. If you have phototrophs, these are microbes that are photosynthesizers. These are going to be your plants or your blue-green algae, your photosynthetic bacteria. Chemotrophs are going to be microbes that gain energy from some sort of chemical compound like glucose. So if you have a phototroph, you're going to gain your energy from sunlight through photosynthesis. Chemotrophs are going to gain energy from some sort of a chemical compound. Photoautotrophs, if we take our heterotrophs and our autotrophs and combine them with our phototrophs and our chemotrophs, we can find out or figure out what their carbon sources and what their energy sources are. Pardon me. Photoautotrophs gain their energy from photosynthesis. They're autotrophs, so therefore their source of carbon is going to be some sort of inorganic carbon, like carbon dioxide. Chemoautotrophs are going to be organisms that use organic compounds for a source of energy and inorganic compounds as their carbon source. You can divide these into lithotrophs. These rely totally on inorganic minerals. And then you have organisms like methanogens that produce methane from hydrogen gas and carbon dioxide, and then they can use that, pardon me again, as their source of energy. You have some bacteria that live in the domain Archaea that are methanogens, and then some of them that live in some very extreme habitats that are also going to be these organisms that are going to be able to produce methane. Majority of our bacteria are chemoheterotrophs, and they are going to derive both their carbon and their energy from organic compounds. Now we can divide these even further, and we're going to talk about one group that are called the saprobes. These are free-living microorganisms. They do not rely on a host. They rely on nutrients from their environment. They feed primarily on dead organic material. Organic detritus or dead organisms. These are our decomposers. You've got a lot of fungi, a lot of bacteria. These are saprobes, and these are going to be those that decompose plant litter. They're going to decompose animal matter, dead microbes. So whatever organic molecule that is available from some sort of dead byproduct, they are going to use that as a source of nutrient and a source of energy. Most of them have a very rigid cell wall, so they were going to release enzymes to the extracellular environment, and this is going to allow for food particles to be digested into smaller molecules. These smaller molecules then can be consumed or absorbed by the organism, and they can then use them for their sources of nutrient and energy. You've got obligate saprobes, which are going to exist strictly on dead organic matter. They tend to live in the soil and in the water, and they can be pathogenic. You do have a number of bacteria and fungi that live in both the soil and the water that can be pathogenic, that are obligate saprobes. You've got facultative parasites that when you have some sort of saprobe infect the host, then that's usually when the host becomes compromised, and this is going to be an opportunistic pathogen. So when the organism is about to die, then these facultative parasites will come in, and they will begin to cause this organism to begin to die. And when that organism dies, then the saprobe will feed on its dead matter, and the facultative parasite then has been instrumental in causing the death of the organic organism. Here you can see the digestion of bacteria and fungi and what they do as they are those saprophytic organisms. So you have here in our first picture here, you've got a bacterium, and you've got this organic debris, whatever it might be, proteins, carbohydrates, whatever. But they can't be consumed or transported across the membrane. So what will happen is you will have these bacteria or these fungi will release enzymes that are going to have an effect on these organic molecules here. These enzymes will hydrolyze these molecules in these enormous pieces of debris into these smaller, tiny molecules. And once they are in these smaller molecular form, they can be transported across the membrane, across the cell wall into the cytoplasm, where they can be used for nutrition and energy. Parasites are going to derive their nutrients from cells or tissues of a host. These are going to be obligate organisms. They cannot gain any sort of food or nutrient from the outside environment on their own. They have to gain it from a host. These are also called pathogens because inevitably they will cause damage to tissues. They can be something very minor to even causing death of the organisms. You have ectoparasites that live on the body, and then endoparasites that live in organs and tissues. So endo and ecto, if you remember what those mean, ecto is obviously going to be outside the cell or outside the body. Endo is going to be inside or living on the inside structures of the body. Intracellular parasites are going to be like viruses. They live within the cell. They're going to then, once they get into the cell, they're going to wreak havoc inside that cell, and that cell is going to die in the end. Then you have obligate parasites. These are unable to grow outside of a living host. A number of bacteria, a number of fungi, viruses, these are all obligate parasites. Matter of fact, viruses are called obligate intracellular parasites because they cannot go through any kind of replication or metabolism outside of the cell. There is no growth outside the cell, and they must have a living host. Once we have all of these nutrients that we have on the outside of the cell, we have to be able to get them into the cell. So we have a number of membrane transport mechanisms. I alluded to a little bit of this in the last unit when we talked about simple diffusion, active transport, that kind of stuff. This is going to be kind of a little bit of the same, but hopefully you'll see how it works in microbial nutrition. Cells are going to have to take up these nutrients, and they're going to have to transport waste out. Just think about when we take in oxygen, we go through metabolism, and the byproduct of our metabolism is carbon dioxide. We need to breathe that carbon dioxide out. If we don't breathe that out, then we have increasing toxins in our body, and a number of our cells and a number of our organ systems will shut down. Just like that, bacteria have to be able to get waste out as well. And so one of the ways they will do that is with these transport mechanisms. Transport is going to occur across the cell membrane even if the organism has a cell wall. Remember that cell wall is like a net. It's not a solid structure. It's hard, but it's got openings in it. And so things can get in and out of those openings. And so the cell membrane is going to be the part of the cell that is going to regulate transport. We have 3 different types of transport we're going to talk about: osmosis, diffusion, and endocytosis. Osmosis is simply the diffusion of water across a semi-permeable membrane. Basically, this membrane is going to have some sort of passageway that is going to allow for water to move in and out of the cell without blocking it and blocking certain other molecules from going in or out. Wherever solutes go, water follows. So if you've got solutes going into the cell, water is going to follow right behind it, meaning that the side of the cell, whether it's the plasma membrane and the extracellular face or the plasma membrane and the intracellular face, whichever side has the most solutes, water will diffuse faster to that side so that it can maintain equal osmotic pressure on both sides of the cell and the cell doesn't rip apart. You have some osmotic relationships. We learned about these all in A&P. Basically, this is going to be a relationship between the environment and the relative concentrations of solutes and solutes and solvents within the solutions on the internal and external parts of the cell. If we're talking about an isotonic relationship, we've got the environment is equal in solute concentration to the internal structures of the cell. You have no net gain of cell volume one way or the other, meaning that the environment is equal in the extracellular side and it's equal on the intracellular side, and there's no net change in the way that water moves. Generally, most stable environment, this is going to be the most stable environment for the cell. If we move to another type of environment, like a hypotonic environment, you're going to have solute concentration that is lower on the external side, and it's going to cause water to move into the cell. And when that happens, the cell is going to swell up and it's going to burst. Think about what happens when you start adding water to a water balloon. Pretty soon you're going to have too much water in there and it's going to rupture. When you've got excuse me, fewer solutes in the external environment than you do concentrations on the inside, water is going to flow to the area of greater concentrations of solutes. So now you're going to have all of this water flowing in. It's following the solutes. Wherever solutes go, water follows. Now the cell is going to swell up and it's going to burst. If we have a hypertonic environment, this is where we have a higher solute concentration in the cytoplasm, in the extracellular fluid than we do in the cytoplasm of the cell. Because we have greater solutes on the outside, water is going to diffuse out of the cell, and it's going to cause these cells to then shrink down, and it's going to be and they're going to shrivel up, and this is something called crenation. These are said to have very high osmotic pressure. That osmotic pressure is causing these cells to shrink up, and they're going to shrivel up and die. The best way to remember this is if I have, iso means the same, so things are the same on the inside and the outside. Think about the solute concentration relative to the outside, to the extracellular fluid. If I have a hypotonic environment, that means that hypo means low, so that means I have low solutes in the external environment. Water is going to move into the cell and the cell ruptures. If I have a hypertonic environment, that means I have higher solute concentrations on the outside of the cell. Water in the cytoplasm is going to move out of the cell to try and neutralize that concentration. And so water is flowing out, and this is going to cause that cell to shrivel up and die. Okay? So hyper, high solutes, hypo, low solutes. This is a good, I love this picture because it shows you exactly what's happening. If I have equal concentration inside the cell to the outside the cell, that means water is going to flow in and out with no problem. Okay, here is a cell that's lacking a cell wall. If we have, this would be a human cell. If you put um, uh, some kind of human cell in an isotonic solution, an isotonic bath, water flows in and out in equal measure and nothing happens to the cell. If I have a hypotonic solution where I have fewer solutes on the outside, greater solute concentration on the inside, water is going to flow in and the cell will actually begin to swell. If the cell has a cell wall, the wall will tend to prevent the cell from bursting. Sometimes if the pressure is so different, sometimes that cell wall will fracture and you will kill the cell. But this is very, very rare. If we have a cell that is lacking a cell wall and we are in that hypotonic situation where we have low solutes on the outside, high solutes on the inside, water is going to flow in and pretty soon we're going to have a cell that completely ruptures. This is called osmolysis, and so you end up basically having this cell rupturing just like a water balloon. Hypertonic solutions, basically what happens is we have greater concentration of solutes outside, lower concentration of solutes inside. Water is going to begin to move out of the cell. This is going to basically cause something called plasmolysis or crenation, where the cell is going to shrivel up. You don't have a whole lot of problem with hypotonic solutions in bacteria because of the cell wall, but in hypertonic solutions what happens is the cytoplasm basically dries up essentially, and everything on the inside has no water source, and so the cell is going to go through plasmolysis, and basically the protoplast or the cytoplasm within the cell is going to shrivel up and die. If we have a cell that has no cell wall, basically the same thing is going to happen. Water is going to fuse out, and pretty soon you're going to end up with a cell that is very distorted, has shrunken, and potentially could be a cell that doesn't have any ability to go through cell reproduction or anything like that. So you could essentially kill the cell through this hypertonic solution. When we have movement across the membrane, this is what is called diffusion. When we have atoms or molecules move in a gradient from an area of higher concentration to an area of lower concentration, this is diffusion. And diffusion across that membrane is going to be determined largely in part by the concentration gradient and the permeability of the substance. So do I have a very high concentration on one side and a very low concentration on the other side? And is my atom or molecule permeable to the membrane? So obviously the greater the concentration difference or gradient, the faster your molecule is going to move across. If that molecule can move across without any sort of aid, then you can have what is called passive or simple diffusion. This is going to be limited to very small nonpolar molecules or lipid-soluble molecules. When we talk about lipid-soluble, we think about something called like dissolves like, meaning small nonpolar molecules are going to be absorbed through the lipid membrane due to the nonpolar nature of the fatty acids within the membrane. Once those nonpolar molecules get past the phospholipid and they get into that fatty acid portion, they're going to then be neutralized, or they're going to be dissolved in that fatty acid, and then the rest of those molecules then will go across the other side of the phospholipid into the internal portion of the cell. We have something called facilitated diffusion that's much like simple diffusion, except it utilizes a carrier protein. That carrier protein is going to bind to a specific substance. Not just anything can go through a particular carrier. Basically, binding is going to cause some sort of change in the shape of the carrier protein, and this is going to allow for transport or allow that substance to be moved across the membrane. Once the substance has been transported, then the carrier protein will go back to its original shape and will be ready to pick up another molecule of that substance. Carrier proteins exhibit specificity. We're going to talk about enzymes and how specific they are. These proteins are very specific. Each carrier protein is specific to its own substance. If you have a carrier protein for glucose, it's not going to take potassium across the membrane. So that carrier protein has its specific molecule, and that's going to be the only molecule that will go across that membrane. You have something called saturation, basically, which is this is the rate of a substance is going to be limited to the number of binding sites. If I have 15 molecules of glucose, but I only have 6 carrier proteins, I have reached saturation, and until some of those glucose molecules get across the membrane, that saturation is going to be maintained, and then eventually I will start having more and more glucose coming across the membrane. This is kind of like standing in line. If you go to an amusement park and you have the roller coaster is fantastic, and you get there first thing in the morning and you're first in line, and it's no big deal, you're getting right in there. But as the day goes on, the line gets longer and longer because there are more people than there are cars or seats in the roller coaster. And until people ride through, the line is going to continue to move and will fill up those cars, and more people will get to go through. Same way with these saturation or these binding sites in these transport proteins. If they're all full, we have to wait for those molecules to go through and the protein to regain its shape before the next group can go through. So saturation is something that you look for in transport proteins. Competition is basically when you have two molecules that have a very similar shape that can bind to the same binding site of a protein. So if I've got molecule A and molecule B, and they look a lot alike and they both fit into the binding site of the carrier protein, it's whoever gets there first, and that's going to be the molecule that gets carried across the protein, or excuse me, across the membrane. So here you have the outside of the cell. You've got all of your molecules here, and then they are going to go in and fit into the carrier protein. That's going to cause that carrier protein to change its shape. When it does, then it releases those proteins to the inside of the cell, and once those proteins are on the inside, it will go back to its original conformation and pick up the rest of the molecules that are on the outside of the cell. So you can kind of see how binding changes the shape. Here it's open on this side and it's closed on this side. So you have a particular molecule binding to the protein. It's going to change the shape from the outside to the inside, and it's going to release those molecules to the inside of the membrane. Active transport is the transport that goes against a concentration gradient. Just like simple diffusion and facilitated diffusion, these are going to go from an area of higher concentration to an area of lower concentration. Active transport does just the opposite. This is like going up the down escalator. This is going to require the presence of very specific membrane proteins. It's going to require the expenditure of energy. Again, you're moving against a concentration gradient. You have a number of items that are going to require active transport: monosaccharides, amino acids, organic acids, phosphates, and a number of different ions. These are going to be things that are going to be in lower concentration in the normal sense, and they're going to flow from an area of low concentration to an area of high concentration. An example of active transport is the sodium-potassium pump. Here is a chart. This is not in your book. This is one that I made. If you look here, you've got your simple diffusion, your facilitated diffusion, and your active transport. If we look at the gradient for simple diffusion and facilitated diffusion, we go from an area of high concentration to an area of low concentration. But in active transport, we go from an area of low concentration to an area of high concentration. So we're just going backwards. In simple diffusion, there is no carrier required, but in facilitated diffusion and active transport, both of them are going to require some sort of carrier protein. Energy is not going to be expended in simple diffusion and facilitated diffusion. Remember, the definition of diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. So if the gradient goes from high to low, then you're going to have no expenditure of energy. But because we're going from low to high in active transport, energy is going to be required. We're going to expend ATP. Because these require no energy, both facilitated diffusion and simple diffusion are both passive forms of diffusion, whereas active transport, obviously because energy is required, is an active transport mechanism. An example of simple diffusion is osmosis. This is the simple diffusion of water across a semi-permeable membrane. Facilitated diffusion, transport of polar molecules. Also as active transport, the sodium-potassium pump. Glucose movement across the membrane is going to be an active transport. Glucose can also be transported via facilitated diffusion. So not all of these are going to, some of them will overlap a bit as far as their examples are concerned. Endocytosis is essentially the eating and drinking of cells, by cells, if you will. It's a form of active transport. Basically, we are transporting large molecules, large particles, lipids, and other cells into the cell. They're going from an area of low concentration to an area of high concentration. This occurs in some eukaryotic cells. For instance, we have white blood cells that go through endocytosis. Basically, what's going to happen is the cell is going to enclose a substance into the membrane and simultaneously form a vacuole, and that vacuole then will engulf the cell. There are two types of endocytosis, phagocytosis and pinocytosis. Phagocytosis, we have amoebas and white blood cells that will ingest whole cells or very large particles of cells that will be consumed and destroyed, and then in the case of an amoeba will be eaten as a source of food, and in the case of white blood cells will be destroyed to keep us from getting sick. Pinocytosis basically is cell drinking. This is the transport of liquids across a membrane, things like oils or molecules that are in solution. Pinocytosis is going to be actively transported across the membrane. Pinocytosis is not going to move water across the membrane. Remember, that is osmosis, and that is a form of simple diffusion. Pinocytosis is going to take solutions or oils, things that are not going to move across that membrane freely, and it's going to take them in through active transport. So here we have phagocytosis and pinocytosis. Essentially, you have these, let's say this is a white blood cell and these are bacteria here. They're going to bind to receptors on the cell, and the first thing that's going to happen after we have this binding is you're going to have the formation of things called pseudopods or false feet that are going to wrap themselves around these bacteria and form this vacuole. Now we have within this vacuole the ability to segregate and destroy this organism. Pinocytosis is basically the same thing that happens, except you're going to have microvilli. We're not going to have any kind of pseudopod here. We're going to use the microvilli, and those microvilli will bind together to form this liquid vacuole. You will also have the movement of oil droplets through the membrane this way. This is going to be kind of a pinocytotic movement of oil. So we have these vesicles that contain extracellular fluids that may have enzymes or things like that in there that will allow for cell functioning in some other part of the cell. You have a number of different environmental factors that are going to influence microbes. Temperature is probably the #1 way to control microbes. Microbes, bacteria don't have homeostatic mechanisms that will allow them to adjust their body temperature or their cellular temperature, if you will. And so, because they can't control this, they're going to assume the ambient temperature of their habitat. And if those temperatures are too high or too low, there's going to be some ill effect on those microbes. You have a number of ranges of temperatures for the growth of microorganisms, and they can be expressed in what are called the three cardinal temperatures. You have the minimum temperature, you have the maximum temperature, and you have the optimum temperature. The minimum temperature is going to be the lowest temperature at which a microbe can continue to grow and go through metabolism. Maximum is the highest temperature at which growth and metabolism can continue, and then optimum is the range where you have the temperature that is going to promote the greatest growth and most efficient metabolism of the organisms. Some of them have a very narrow range, while others have a very broad range. You have some very fastidious organisms. Neisseria gonorrhea has some very narrow minimum, maximum, and optimum growth temperatures. But if we look at something like E. coli, it can grow at room temperature, it can grow at body temperature, and it can even grow a little higher than body temperature. So it has this very large range. Another way, excuse me, to express temperature adaptation is to express whether the organism grows at cold temperatures or moderate temperatures or at hot temperatures. And these are going to be what we call the psychrophiles, which like it cold, the mesophiles, which like it moderate or in those middle ranges, like body temperature, and then thermophiles, which like it hot. If we're talking about a psychrophile, this is a microorganism that has an optimum temperature that is below 15 degrees centigrade and is capable of growth at 0 degrees centigrade. So these are going to be the organisms that grow in your refrigerator. These are the things that make things go bad in your refrigerator. True psychrophiles are obligate with respect to their temperature. They can't grow above 20 degrees centigrade. So that is pretty chilly. Psychrotrophs or facultative psychrophiles, these are going to grow slowly in the cold, but they have an optimum growth temperature that is above 20 degrees centigrade. So most of the organisms that we will use in lab are going to be these psychrotrophs or facultative psychrophiles. It's kind of hard to keep true psychrophiles growing and maintain them in the laboratory. So the ones we will use will be those psychrotrophs or facultative psychrophiles. Here you can see on this picture, which is snow. There's snow here, but you've got this red or pink kind of tinge or tint here in the snow, and that is this red organism up here. This is one of those, you'll see here, these are little ice crystals, and this is an organism that grows very freely in the snow, and this is one of those obligate psychrophiles. This picture is in your book. I don't remember exactly which page it's on, but it's in your book, and you can look at it a little more closely. Mesophiles, meso means middle, and these are organisms that are going to grow in those middle ranges or intermediate temperatures. Their optimal growth temperature is going to be between 20 to 40 degrees centigrade. So 37 degrees, which is body temperature, sticks right in the middle of that. Most human pathogens have an optimum growth temperature between 30 and 40 degrees centigrade. So most of our pathogens are going to be mesophiles, and because that internal core body temperature is just right in the middle of their optimum growth temperature range. Thermophiles, these are going to be microbes that grow optimally at temperatures that are greater than 45 degrees centigrade. They're going to vary in heat requirements. Some of them are going to be extreme, and they grow between 80 and 120. Some of them have a very general range that's going to be between 45 degrees centigrade and 80 degrees centigrade. So we're going to actually use one that grows best at about 55 degrees centigrade when we start talking about the temperature ranges of bacteria in lab. I think that's actually lab number 12 that we're going to be looking at. So if it's not, it's right close after that. Just like nutrient and temperature, Bacteria also have specific gas requirements, particularly oxygen and carbon dioxide. Atmospheric gases are going to be very influential on microbial growth. Does it use oxygen? Does it use carbon dioxide? Oxygen has the greatest impact on microbial growth. We can have some bacteria that, as oxygen enters into cellular reactions, it can transform it into a number of toxic products. Sometimes we talk about these, trying to remember what they're called now, free radicals. People that smoke, once oxygen gets into the cell, it is actually going to be converting some of that oxygen into free radicals, and that can lead to some of the things that we see as a byproduct of smoking. People who tan, the same thing happens, and that's where you get your wrinkles and things like that. Most bacterial cells have developed a set of enzymes that will scavenge these free radicals and neutralize them using a couple of different enzymes, one of them called superoxide desmutase and another one called catalase. And again, we're going to start looking at some organisms in lab that make these enzymes, and we'll be able to see the conversion of that oxygen into a usable product. These enzymes are going to be essential for bacteria that we call aerobic organisms that require some sort of oxygen in their environment for growth. You have a number of different categories of oxygen requirements. We're going to talk about them all here. Let's first look at the bacteria that require oxygen. These are aerobes. They can use gaseous oxygen in metabolism. They also possess enzymes that can break down those toxic oxygen products and make them into usable oxygen. Obligate aerobes cannot grow without oxygen. If they don't have oxygen, they're not growing. You are an obligate aerobe. If you don't have oxygen, your metabolism ceases and you will die. Microaerophiles, these do not grow at normal atmospheric oxygen, but they require very small amounts of oxygen in their metabolism. Micro means small. If we are an aerophile, we are oxygen loving, so we like small oxygen. So these microaerophiles are going to use small amounts for metabolism. The anaerobes, these are not going to require oxygen, and there are a number of different anaerobes. They lack the metabolic enzymes for the use of oxygen in respiration. One of them is called a facultative anaerobe, and this is an aerobe that does not require oxygen for its metabolism, but is capable of growth in the absence of oxygen. So that's why they call it a facultative anaerobe. It is an aerobe by nature because it can use oxygen, but it doesn't require it, and it is capable of growing without it. Strict or obligate anaerobes are just like obligate aerobes. They cannot grow in the presence of oxygen. They cannot tolerate any free oxygen at all. If they are exposed to it, they'll die. They don't lack the enzymes to process toxic oxygen, so those toxins build up in the cell, and the cell then will eventually die. Aerotolerant anaerobes, they don't utilize oxygen, but can survive and grow in limited amounts of oxygen. They don't use it. If you expose them to too much oxygen, they're going to be like the obligate anaerobe, and they will die. They have very limited ability to break down those toxin-oxygen products. Here we have a set of test tubes. This picture again is in your book. I have labeled the tubes. The tubes aren't labeled in your book. But if you look here, we have an aerobe. This aerobe basically has growth only at the top. If you look, the media itself is clear. You have a facultative where you've got growth. This is going to be one that probably requires a little more oxygen than just the generalized facultative, because you see that this is staying mostly in the top half of the tube. Here is a true facultative. It grows with or without. You'll see that the tube is completely filled with bacteria, and this organism is growing with or without the presence of oxygen. And here is an anaerobe. You'll see that the top of the media is completely clear, and the closer you get to the top, you'll see that you have fewer and fewer dense areas within the test tube. And so it's going to be staying down towards closer to the bottom of the test tube. Carbon dioxide is required by all organisms in their metabolism. They are going to have some kind of carbon dioxide, basically. What we call capnophiles, these grow best at higher concentrations of CO2 than you normally have in the atmosphere. So when you have these organisms and you're incubating them in the laboratory, you have to put them in a high concentration of carbon dioxide, or they don't grow very well. If you're going to be isolating certain pathogens like Neisseria gonorrhea or Brucella or Streptococcus pneumoniae, these are all capnophiles, and they are going to need higher concentrations of carbon dioxide. They're also going to take generally a little bit longer to grow than your normal bacteria. When we use Streptococcus pneumoniae in lab, we generally have to start it a couple of days before we start the other stuff so that it has an opportunity to grow. pH also has an effect on bacterial growth. The majority of organisms are going to live best at a pH between 6 and 8. Most of them live the very best at a pH of 7. But you do have bacteria that do like to grow at lower pHs, and these are called acidophiles. These are going to be those organisms that are going to like it at a relatively low pH. You have Euglena mutabilis. This is an algae that is going to grow between a pH of 0 and 1. This is the pH of your stomach. Then you've got thermoplasma. This is an archaea that lives in hot coal piles, and the pH of those hot coal piles can reach as low as 1 to 2. Basically what happens as the higher the pH gets, the cells will begin to lyse, and if it's exposed to the pH of something like 7, you will completely kill the population of cells. Alkalinophiles, these are going to be those cells that like it at a higher pH. These are going to be the opposite end of the spectrum. Natropnemonas, this is going to live in hot pools. It's also in the soil, and it can live in very high pHs, pH of 12. The pH scale only goes up to 14, so you're talking about something that loves it extremely, extremely basic. The Proteus species, this is going to use something called ammonium, which is basically the ammonia ion, to neutralize the acidity of urine. And when it neutralizes that acidity of urine, it can then colonize the urogenital tract and cause UTI. So this is a huge pathogen that can cause significant urinary tract infections, as well as some urogenital infections of some of the upper parts of the urogenital tract. Ecologically, there is some association among microbes. Most of them live in shared habitats. You don't have isolated pools of bacteria here and isolated pools of bacteria here. They're going to form these shared habitats. You're going to have interactions between these organisms that can be beneficial, they can be harmful, or they can have no particular effect on the organisms that are involved. You can have these relationships that are obligatory, meaning that they are required to have this relationship for survival, or they can be non-obligatory to the members in the relationship. They can take it or they can leave it. They are going to involve generally some sort of nutritional interaction. When we talk about this, we're talking about something called symbiosis. This is going to be the generalized term that is going to denote some sort of situation where you have two organisms that live together in a close relationship. When we talk about the organisms or the members in these symbiotic relationships, they're called symbionts. These symbionts then can display three different types of symbiosis. The first one is called mutualism. Mutualism is when organisms live in an obligatory or mutually beneficial relationship. They require each other. Each of them gains some sort of benefit from that relationship. Commensalism, this is where a member is called the commensal where the member that is called the commensal is going to receive the benefit, where the co-inhabitant is neither harmed or benefited. So you can have relationships where you have one organism that is obligated to have a relationship with another organism, but that other organism isn't harmed or benefited at either way. Satellitism, this is where one member provides nutritional or protective factors that are going to be needed by the other. Think about our gut flora, our E. coli in our gut. We have a commensal relationship with that E. coli. It provides provides our vitamin K, which is going to be some sort of nutritional factor that is needed by us. That E. coli is gaining no benefit or no harm from living in our gut. Then the other symbiotic relationship is parasitism. This is a relationship in which the host organism provides a parasitic microbe with nutrients and a habitat. The host is gonna have harm. A parasitic relationship is going to cause harm to one of the symbionts in that relationship. The parasite itself that parasitic microbe is going to gain all of the benefit, and the host is going to be the recipient of all of the harm. So this would be what we call a plus-minus relationship. A commensal relationship would be a +0 relationship, and then mutualism would be a plus-plus relationship. Non-symbiotic relationships are going to occur in organisms that are free living. They don't have any required host or cell-to-cell relationship. These are going to be what we call a synergistic or antagonistic relationships. Pardon me. Synergism basically is an interrelationship between two or more free-living organisms where the relationship benefits them, but it's not necessary for their survival. Okay, so this is, you're going to have what we call a synergistic effect. But if one of those organisms leaves the relationship, the other one is not going to be harmed by the exit of that organism. Antagonism is an association between free-living species that are going to arise when members of the community are going to compete with each other. One microbe is going to secrete some sort of chemical substance to the environment that is going to inhibit or destroy microbes in that same habitat. This will allow that organism to have greater access to food and to space for microbial growth. You have bacteria, the Streptomyces species of bacteria and the Bacillus species of bacteria have antagonistic relationships with other organisms because they secrete chemical substances that we have purified into antibiotics. And those antibiotics then that are secreted by these organisms inhibit the growth and metabolism of organisms in that same habitat. Normal biota basically is that normal flora that lives on the skin and the alimentary or the gut. It also is in other sites of the humans, in the eyes, in the nose, in the mouth. We have it everywhere that we can have a space. There is bacteria. These can be commensal relationships, they can be parasitic relationships, and they can be synergistic relationships.